An ecosystem is a slower responding issue than the news cycle. Ecosystems can take years to show damage, but that also means they take years to recover from that damage. An example of this that we're seeing is the Exxon Valdez. We're still seeing 5-10 years down the road, there's still issues that are coming up new to us about the ecosystem. We're still finding out that the oil is still there and it's decomposing in different ways. So this is part of what we want to do, is look at the geochemistry of the system and look at the chemistry of the system itself to find out whether that system's changing with the oil coming in. So we can try, hopefully, detecting oil by some of our advanced techniques. But if we don't, we can see, does the overall signature change? And how does that overall signature change? By knowing how that overall signature changes, can we then link that back to the damage that was done?
And the change that you're looking for is a more subtle change than, for example, all the fish dying and not coming back. You can...
Yes. Basically, what a lot of people have seen on the news is a macroscopic point of view. You see a fish floating, it's dead. This is a very clear, okay, that's bad. Pelicans covered with oil, okay, this is bad. But if you remember, they had a lot of turtles come in that were dying, and then, well, was it the oil or was it not the oil? And we still don't have a very clear answer on some of these issues. With the oysters, another example, are they healthy for me to eat or are they not healthy for me to eat? That's a macroscopic one. We're much more interested in the microscopic ones about basically, has there been a long-term perturbation to systems so that the ecosystem's not as healthy? An example about this, again, is if I shoot you, if you get shot and you die, that's a very quick response. However, if you have cancer, it's a long time before we see that, and there's a long degradation, and things change. Similar to most illnesses with people, we usually see, for some people with Alzheimer's or something, a change in their personality. But the disease has already been in there for a long period of time. We want to find out whether there has been this type of scarring of the ecosystem that's that deep, that's going to take for a long time for it to recover, and what those symptoms will be, and this is one way of monitoring them.
So how many times have you sampled already, and how long will you sample?
Okay.
Ideally, or based on funding or whatever.
Okay. So what happened was the oil spill happened, and we all saw that on the news. I think we all sort of saw it that, initially it was okay, it's a blowout, and then maybe 2-3 days later it sort of came out, well, there's a oil leaking in, and then a few days later it's like there's a lot more oil coming out than we thought. At this point in time, I and two of my colleagues, again, Mike Perdue and Tom Bianchi, got together and talked and said, what can we do that would be unique? And that was the marshlands, not the deep water, but the shallow water where the U.S. and a large amount of us get our seafood from. And these are very delicate ecosystems, as Hurricane Katrina showed as well, that are very damaged already. What happens when you actually add another injury to an injured system? So what we're trying to do was we're trying to get together that. So what we did is we applied to NSF, National Science Foundation, which is the part of the government that basically funds a lot of this basic research in the nation that we rely on to do more advanced analyses. And it was through what they call their RAPID program. And they actually have one that they can actually do a very fast turnaround to give you money for situations like this. So the RAPID grant that we got lasted a year. We've been out and sampled one time up to now. We're going out and sampling the next week or two. And that just depends on schedules as well as weather as well as flows. And on the first sampling, we did what was called Barataria Bay, which again is a very important bay and has a large amount of bio importance for the state. Next one, we're going to do Barataria, and then we're going to do a bordering one called Terrebonne. The reason we want to do these two is we found out the Barataria, what they'd actually done is they allowed Mississippi water in through what they call the Davis Pond mechanism, and this is basically put a positive pressure of fresh water at the front end, so the fresh water just flushes similar to a tube, so it outcompetes the Gulf water coming in as a way of keeping the oil out. Terrebonne, that had not been done. So now we have two comparisons between two systems that had been treated differently. Can we see differences in the recovery? That's the next sampling we want to do. The RAPID grant that we have funds us for a year. We're now going to be applying for more money. And I just want everybody to understand that. We work on granting, and the federal government gives us grants. Each time we do something, we have to write a whole new grant. It then goes through this whole peer review process where people very critically look at this to make sure that that money is being well invested in. So this basically means that we now have to, although we have the money, that does not mean we're going to get automatically the next set of money. We now have to apply and compete again. So this is a very highly competitive, very good way of keeping everything competitive. So we want to sample as much as we can, but as well, sampling takes money, and we only get a certain amount of money, so we have to be very logical about how we're doing this. This is why we want to sample now. It's a distance away from the oil spill after the oil's been switched off and everything to find out what's happening. And we'd like to possibly sample again if we can. And again, we'd like to do this for maybe 3-5 years if the funding's available and we're getting results that support us to continue on with that. Okay, so when we got the RAPID grant, it allowed us to actually go out in the field and collect water samples very quickly after the oil spill. So we basically had as close to a starting point as possible. The reason this is important is we want to know how that ecosystem looks at that point in time, because that allows us to see how that ecosystem would recover. So that was the first part of the grant. The other side of the grant was for us to go into more detail in the chemical, and I don't want to hurt anybody with the minutiae of all the chemistry that thrills me, but it doesn't thrill other people. I know that other things don't thrill me, but basically the idea is the grant gave us the ability to do that. The need is that we need to understand the ecosystem. The grant gives us a tool to actually do our research as one prong on approaching that. Now, in no way does this give us all the answers, and in no way does science, each project, give you all the answers. It's a combination of these different projects. Our grant, we're using a multitude of different methods, or a number of different methods, to try seeing if we get a convergence. Or basically, do they all come to the same type of story? Again, if we go back to interviewing or interrogating, the police officers will come back with another question that may be half an hour later to make sure that you're giving a consistent answer. We're doing the same thing with the different methods. Do they give us a consistent answer so that we can actually understand the system? What we're going to get, we don't know, because research takes a long time. The water that we processed, we collected, as I said, the 2 metric tons of the water. It was brought here. In 2 weeks, it was reduced down to around 20 liters of water. So that's a good factor of 100 that we've reduced things down. Now that water needs to be freeze-dried. So for everybody that actually drinks coffee that's freeze-dried, just an idea is basically what you do is you basically freeze the water and then you dry it out without it ever going back to liquid state. So it just goes into the vapor state under vacuum. This takes a long time to process this amount of water correctly without an error happening, because in that stage we cannot have it thaw out. Once we get that, we then get a brown powder that will look like chili powder. It won't smell like chili powder. It may smell a bit like soy sauce, to be honest. We get this brown powder. Once we get the brown powder, we then put them into our instruments. And once we get the data from the instruments and combine them and compare them, only then can we really give a detailed story. Similar to only when the doctor gets your cholesterol, your lipid levels, your glucose levels, can they really tell you what type of dietary changes you need to do. So
with respect to the salt marsh, when you dry out, you're collecting water not actually in the salt marsh, you're next to it, in the water that's going to flood the salt marsh and the water that's going to drain the salt marsh. So you do have information about the salt marsh, but what are your data going to say about the salt marsh?
Okay. When we collected samples, basically there's a salt marsh, which is basically just the soil and grass growing. We collected as close to there as possible, usually within 10 feet. So 3 meters. And what we wanted to find out from that is what the water's like, and is the salt marsh dying off and releasing material. Similar to what my friend and my colleague Tom Bianchi says is like a tea bag. Exactly like a tea bag. You put a tea bag into boiling water, you sort of notice there's a nice concentrate area, then it diffuses out. That's what we want to find out from the salt marshes. We want to find out whether the oil contaminations cause damage on that, so it actually releases different signatures than the non-contaminated. An example of this is if plant material is dying. As we know, if you cut grass, it sort of rots in your yard. Well, does we get that type of rotting? By that type of rotting, it's releasing different chemicals in there that will give us different biomarkers, that will give us different signatures on our spectroscopic methods than, say, a healthy one. And this will allow us to know whether that's breaking down or not. And then what does that mean in terms of the chemistry, overall chemistry of the ecosystem, now that we've actually had an increase of input into biomarkers or biocompounds A versus B?
And what's interesting is that the way that it rots is important. That's what you're seeing, is the way it rots. But the rotting vegetation is part of the food web. And so if it rots differently from what we would expect in a healthy situation, that could be a problem.
Okay. Well, when plants are rotting, what are actually happening is they're releasing a lot of material into the ecosystem. Some of that can actually be used for food sources. An ecosystem is a very fine balancing act of multiple points. This is not you on a teeter-totter. This is you on a teeter-totter, 3 dimensions, with tons of other people on it and tons of other points. If one of them gets pulled down, it may affect this one, that may affect this point. So when we're doing this, this may actually favor one type of organism in the ecosystem over another, and that may then unbalance the ecosystem and do a debalance of the ecosystem where the ecosystem may not be able to come back. Worst case scenario would be if it actually favored an invasive species or a species that's not native that has somehow gotten into the ecosystem through flushing of boat tanks or something. That's one. The other one is that the chemistry of the natural organic material will now have changed. So that means how it transports the oil and other nutrients will have changed on top of that. So it's not only a food source, it's also the transporter of other food. So it's sort of like the hot dog bun with the hot dog inside. And different hot dog buns, as we know, you can put different condiments in. And, you know, sometimes you get a hot dog bun that just goes and goes mushy in your hand. Other times it stays nice and firm, especially if you put the pickles and so forth on it, you know, the Chicago hot dog. Same idea here. If we basically start losing or adding different chemical components in there, it can change the food web because of the way it changes the dynamics of how that food's assimilated by different organisms. Within this, a lot of us look at fish and go, okay, that tells us the health of the ecosystem. But that's not true. Others of us look and go, the water tells us the health of the ecosystems. That's not true. It's a combination of everything. It's like when you eat, you just don't drink. It matters about the food you eat, the drink you drink, how you drink it, how you eat it, and what you drink, and whether it's bubbly or not, does it have sugar or not, do you drink lots of alcohol or not. We all know this when we go to the doctors. They are asking us these questions about our dietary. Same thing for the ecosystem's health is this dietary. And if we change something in that dietary process, we can change the ecosystem. Society is an ecosystem itself. We can look at it that way. And as we basically change the diet within Western or especially American areas, we begin to see that sometimes our children are getting larger. I don't want to use the word obese, but larger than we're normally used to doing this. And is this because they have less activity or because we're changing the food structure? And is it because we're changing what we're putting in the food? So these are some of the ecosystem problems that the ecosystem also responds to. So just by looking at one component is not the best way of doing it. The problem, however, is a fish, as I said before, it's dead or it's alive. This is a very simple 0 or 1 type process. However, if the fish is unhealthy, is it unhealthy, and when does it get unhealthy? And how do we monitor that unhealthiness of a fish? It may swim correctly, it may eat correctly and everything, but it may have a tumor on it. Similar to we've seen this with athletes in professional sports and everything. Similar to some people in your family when they go to the doctor and they may get diagnosed with a disease and you're going, but they're perfectly healthy, doctor. I mean, same thing happens with most of us. I mean, you know, your cholesterol's high, but I'm perfectly healthy. And only when we get down to that molecular level do we get this. So our grant's trying to take that within the shallow water or in the salt marshes, which are very delicate, unique ecosystems in Louisiana in the Gulf Coast. And the reason that there's a lot of focus on Louisiana sometimes is because that's where the Mississippi River that produces a lot of these are. But the whole Gulf Coast itself is an integral part of the whole thing, so we can't just only focus on one area. So this is one of the issues we have, and this is one of the problems that we have, and NSF and everybody has, is granting everybody to do all the work they want. And I'm sure from the rest of the program, you're going to see scientists doing a whole range of things. And a lot of us do talk. We'll have arguments, we'll have debates. We'll have friendly talks. Just because a scientist disagreed does not mean either one of them is wrong. They can both be right. It's just where do they have that overlap of being right? And this is sometimes where we sometimes have to do adjustments to be able to bring the numbers together. And something that we worry about is the general public sometimes sees that as us trying to force an issue. No, it's just us trying to bring basically within legislation. How can we bring two legislation pieces together while maintaining the spirit of each one? So this is sort of what we're doing, and we sometimes, we're humans. We use slang language. We sort of joke around in emails and everything, and we hope that doesn't get misinterpreted. And sometimes we'll really prod somebody and go, yeah. But as a whole, we do get along. We do want to get along, and we do share data. On this project itself, Dr. Bianchi is an oceanographer. I'm in the chemistry department, and Mike Perdue's in an earth and atmospheric department. But the three of us get along very well. We can write papers, proposals, and everything together. We also have Gary King, who's doing some of the microbial stuff with us that we want to bring in the fold. So basically the idea is we want as many people, as much data, and then we basically, we'll have arguments about what that data means. I mean, how many times have you actually gone out with your family that have all agreed to one restaurant? You'll have an argument, you'll have a debate about it, but that does not mean the family does not get along and that anybody's wrong. I mean, all the restaurants can be good. It's just that, you have to decide on one. So Louisiana is sort of a unique-ish state within the U.S. We're known as a state without beaches that's on the Gulf Coast, but that allows us to have a series of salt marshes, wetlands, and everything that are extremely rich in terms of the diversity and the food that comes from them. So a large amount of the mussels, shrimp, such types of food come from these, and they're basically very important incubators for a lot of the Gulf system as we know it. On top of that, the wetlands actually act as a filter, so they clean the water before it hits the Gulf of Mexico. So when you come over to Louisiana, one thing you're going to notice is you go over a lot of swamps. And first of all, I mean, I came down here, and I'll be bluntly honest, I came down here from Canada. And swamps were very new to me, and I thought, oh boy, what does this all mean? Once I got into swamps, I sort of saw the amount of fish, the amount of food, the amount of just biodiversity and richness within those, such as how many people like blackened catfish? Well, we had the catfish. We also have the shrimp. We have the mussels. We have clams. We have all sorts of different fish, redfish and so forth, that do use the Mississippi. The Mississippi does deliver a lot of nutrients down into the Gulf. And although Mississippi and Alabama have been very lucky, Texas has also seen some of the bad effects of this, where the Mississippi has basically been steered down by a bunch of what I'm going to call a pipe. It's basically a pipe that's open on top. Basically, you steer the water down, and this basically means that it doesn't get to go through these kidneys or these filters. And this basically means that we basically dump very rich, nutrient-rich waters into the Gulf. This basically means nutrient-rich waters. By that, I mean basically we provide nitrogen and phosphorus in these waters. Nitrogen and phosphorus are some of the limiting nutrients in the environment where things usually have to work very hard to get them. They develop them. If we basically give a humongous pulse into them, that means is they gorge. Similar to us at a buffet. And that gorging then means that they die after a certain while, and then they decompose, and this is called the dead zone, because basically this causes oxygen. So Texas has sort of seen some of this. Mississippi may not have. On the other side, Mississippi does have some swamps and some wetlands as well, where some of this diversity is. But sort of Louisiana is unique in the fact that we have the pipeline of the Mississippi going through us, and that's been steered, so we've not been having this replenishment. So we've been losing a lot of wetlands within the area, and that's also damaged the wetlands. And Hurricane Katrina affected both states heavily. We know that. It basically did a direct hit on Mississippi, but also it did some very strong damage to Louisiana, especially the New Orleans area, because we lost those wetlands, so that storm surge went directly into us. And this is some of the things that we're looking at in much larger problems, is those wetlands and what they do with the barrier islands and everything to protect us. And also when we lose that protection, we lose that ecosystem, we lose that food. And I don't know the numbers, but I know that we make this whole area, the Gulf Coast area, Louisiana, has a large amount for energy and has a large amount of that seafood that the rest of the country eats. And basically this seafood, by these type of environmental damages, gets threatened. And I think we've seen this. And I know that a lot of Mississippians, I think, like some Louisiana food. I sort of been to Mississippi, and I sort of knows Tabasco sauce and as popular in Mississippi as it is here. So that's sort of the difference. And the states are close in some ways. They're different in other ways as well. But basically it's that ecosystem that we all want to protect because of the value it is to the nation.
Okay, and so when this oil blowout happened, the spill, the oil everywhere, how did that change your research from what you were doing before this all happened?
Okay. So our research program for my own research group is we basically break it into three parts. We do soil, that we call the solid-liquid interface. We do biomembranes, such as blue-green algae and mussels. And we basically look inside and see how their life cycles are affected by different toxins. And we do water. And we've done water in the Chafalaya Basin and so forth. And we're mainly dealing with fresh, not salt water, because of this isolation method. This changed our focus completely to saltwater marshes. So before then, we never really looked into saltwater marshes. We sort of said that's too difficult. This now meant that difficulty was supplanted by need. And it was decided that at this point in time, we would work with other people as well to get this to work. We had done things with Katrina, so this is another way that we knew Dr. Perdue. When I'd asked him if we can send him some Katrina water because it was saline as well to use his method to isolate, he said yes. But usually our group as a whole does not do a great deal of water until recently, and this is basically pushing us into a new direction that we're very happy to go into.
Okay.
When I sort of look at this disaster, one of the questions that keeps on coming up to me is, how long is it going to take for the system to recover? Where is the oil? What's happening with the oil that may be deeper in the water column than we traditionally look at? And how long will it take for the system to flush that out of it, if it will? So one of my big questions is, we may say the saltwater marsh is fine, it's fine, it's fine, and then all of a sudden, due to currents and movement, we may get fresh oil coming in. An example of this that the general public sort of saw during this blowout was there were some tar balls on the Keys in Florida. And then it all suddenly came and said, this was not BP blowout. Well, where did that oil come from? It may be quite old. It could be in the system a long time. The other one is, we know naturally there can be some oil releases. How does the system take care of those? What are their volumes versus this volume? And how did we do differently here with the dispersants? So the big question is how does this, if we look at it, and this is going to not go across well to everybody, but it's been done, it's happened. Can we actually get as much information to help ourselves and enrich ourselves? So we want to look at this and now say it was an accidental experiment. What can we learn from this experience to help us out more than just going, it happened, close our eyes? No, we need to understand, in case something like this happens again, what's the correct way of handling it. I mean, in all honesty, we hope this never happens again. We hope that they basically learn lessons to actually shut it off before any oil ever comes out. But we sort of saw during that oil spill, the coverage, there was another oil spill where somebody sheened off one of the, I forget what it was, but basically we had this thing going up 150 feet up in the air. I think they sheened off a head. We had another oil spill up in the northern states within a river. So we do have oil spills. Very often we need to know how nature's going to process them under different environmental conditions. This is a large one. We've had other large ones in the Gulf War that took place in that area. We've had another large one up in Alaska, and we have this one. And basically, can we learn how they work similarly and how they do work differently and why? So in a quick one sentence, the big question is what are the long-term effects and how long will they be there?
Okay, that's very good. Are there any, I think what Jesse was alluding to is, are there any questions that you have that are very compelling to you, whether that be within your specific field or discipline or something else completely different about this oil spill that you think is just an interesting, very interesting topic that you'd like to know more about?
Well, two or three of the issues where it was sort of fascinated me to actually hear people say that from images they could actually figure out the flow, and others said they couldn't. That technology, I think, needs to be developed much more, especially if we do more underwater exploration and harnessing. This is a mile deep. Humans can't go there. I think that became very clear in the news. We have robots doing all this. We're not going to go into blame gaming and other things, or why did you be able to do that, but you can't figure out how to recover it. This is something that, most of us always get told, don't do something unless you can fix it. But on the scientific side, one of them is what happens under those pressures, under those temperatures down in there when we start adding dispersants and everything. Because that is a very different new area under those flow rates, with those hydrates, with the temperature, with the pressure. What is that? How can we basically develop things to actually understand that? Number two is, in the long run, how quickly can the system recover? And does our system basically have a naturally built-in defense? And the last one is basically within oil you have a mixture of chemicals, which not many people discuss. They basically said you get these oil balls that move. Well, the oil ball doesn't move. There's a bunch of chemicals that have different solubilities and will move differently, so that will be separating at all times. And how do we locate those, trace those to find out what's moving and what's not and where it's going? So it's not going to be a homogeneous chunk of oil. It's going to dissolve, similar to how when you put pills in. I don't know if anybody's done gobstoppers or those little balls that used to have different things that used to dissolve at different rates in your tongue. That's sort of the golf ball, right? So different types of molecules are going to come off, which basically means different colors or different tastes are going to come off at different times. And that's something that I'm sort of very interested to see how that unfolds.